Hello, everybody. Let's get going. Before we start, we've got a midterm coming up in a week. It's going to be very similar, pardon me, in design to the last one. In terms of number of questions, it'll be mostly multiple choice, be some short answers. We haven't settled on absolute numbers yet, but about the same length. It'll be again for 24 hours, open for 24 hours. You'll have 90 minutes to write it, but at any time during that 24 hours. It'll be covering the material from the last midterm. There won't be any questions about the stuff covered in the first midterm, and not specifically about them. We do assume you've assimilated that material. While we won't be asking you directly about it, we will be assuming that you know it. So bear that in mind. Now we'll put a breakdown up on D2L on probably on Monday. Well, certainly, yeah, it'll have to be Monday. So you'll know how many questions will be devoted to each of the topics we've covered. And really, most of this one is going to be plants. The cutoff for the material to be covered on the midterm is on Monday. At Monday's lectures, the last material that'll be covered or liable for the midterm. That's only 4 animal lectures, so the bulk of it will be botanical. Now, you know where to reach me, or you know where to reach Mr. Hickerson if you want to go over any of this material, so don't hesitate. And I'll have more to say about it when the date gets a little closer. Okay, right now we're still putting it together. And the third tutorial will be opening up on the 2nd of November, which I believe is Tuesday. It'll be on transition to land, and this one will have 2 papers. There'll be one on plants and one on animals. So be on the lookout for that. Now, do we have any questions? Will there be practice questions? No, not this time. You've seen the sorts of things that we're liable to do. So you've had your practice. And the tutorial will be due on the 15th, yeah. I believe that's after reading week. So there's something to do on during reading week. I mean, otherwise you'd empty days. Okay, let's get back to animals and osmoregulation. And if you'll give me just a minute here. So here we were on Monday. We'd figured out, looked at osmosis, the tendency, and I defined it pretty broadly, the tendency of water to cross a selectively permeable membrane. Now that's critical, okay? There's got to be a selective permeable membrane between the 2 volumes of water in order for osmosis to take place. Otherwise, you just get diffusion because the 2 volumes of water differ in their solute concentration. And the membrane being a semi-permeable membrane, it's impermeable to, and the selectively permeable and semi-permeable are synonyms. The 2 are used interchangeably. I'll try to stick to selectively permeable, but I grew up with semi-permeable. Anyway, selectively permeable membrane is impermeable to, in this case, the glucose. So glucose can't get through there, but water can. And just to, well, we went through this on Monday, but just one thing to stress. We never tend to think of it this way, but what's happening here is water is diffusing down its concentration gradient, okay? The concentration of water here is less than the concentration of water here. This is pure water, there's nothing else there. Concentration of water is very high. There's these glucose molecules taking up room here though. Note that the volumes initially are the same. So these glucose molecules are taking up room that could otherwise be taken up by water. So the concentration of water is lower here than it is here, and like any substance, water will diffuse down its own concentration gradient. So we always think of diffusion as being a matter of solute dissolving, or sorry, going from where it's more common to where it's less common, going down a concentration gradient. But the opposite is happening at the same time, even if there's no membrane in the way. Okay, if we just had a tube here with no selectively permeable membrane in the middle, and we put in glucose molecules, dissolve them in this half, well, they're going to diffuse down their concentration gradient because the concentration initially here is going to be high, and they're going to diffuse in this direction because they're rarer that way. But reciprocally, water, being more common here initially, is going to diffuse in this direction as well, in the opposite direction to the glucose. So the diffusion of a solute through a volume of solvent is matched by the diffusion of the solvent into the area where it's less common, where the solute is more concentrated. Okay, this is from the, let's get rid of this. This is from the...
Professor, we have a question.
Let's hear it.
So if solutes take up some of the volume, does this mean that if you dissolve a solute in water, that the total volume of solution will increase?
Well, yeah. Usually not, sorry, not noticeably, but yes, it's bound to. They're taking up room. I think you'd have to have a pretty good eye to spot that, but if with the proper instrumentation, you could see it. Okay. But we're disregarding that here. Okay, it's not an important part of the relation we're looking at. Anyway, here's another U-tube with a selectively permeable membrane, and there it is, or semi-permeable membrane, sorry. And it's dividing two volumes of water, pure water, okay? So there'll be a net movement of water. Well, there won't be any net movement of water here. You're gonna get water diffusing in this direction just as much, or an equal quantity of the water diffusing in that direction, okay? These things are always dynamic. There's always something going on, but the two diffusions cancel each other out. Now, the osmotic potential, and I'm going to talk about this with regard to animals. There's a lot more to be said about this with regard to plants, and I'm going to leave that to Mr. Hickerson. Okay, I don't want to get into it in plants because I think you should be getting it from the word go, from scratch. Okay, so I'm just going to give you some basic concepts: osmotic potential, water potential. And let it go at that, okay? Because those are relevant to animals. So osmotic potential, and that's what we call it in animals. It's called solute potential in plants. It's the force exerted on water because of differences in solute content across the semi-permeable membrane. So here we have none, okay? There's no osmotic potential here. There's no difference in solute content. Here, somebody is dumping some solute into one side of the tube, okay? So we're getting osmotic potential being generated. The pure water here has an osmotic potential of 0, none. And that's the highest osmotic potential possible. The lower osmotic potential is a negative number, and that's what you get with solute. And the more solute that's dissolved in the water, the more negative the osmotic potential. Now, water is going to move from an area of high osmotic potential to an area of low osmotic potential. We see this happening here. So we're getting this osmotic movement. So we can define two volumes of fluid in terms of osmotic potential. Do they differ in solute content? Are they separated by a semi-permeable membrane, such as a cell membrane? Then we're going to get net movement of water from the area of the higher osmotic potential to the area of lower osmotic potential. So ignore this for the moment. Blasted, where's it? Yeah, there we go. Ignore these for the moment. We're just talking about this. This is where we're seeing movement of water from an area of high osmotic potential to an area of lower osmotic potential. And you can see that the column is increasing in height. Okay. Now I talked about, actually, I'm just going to leave that. So we all okay with this? No questions? Okay, there's our figure again with a semi-permeable membrane. And what we're interested in is this half now. And that illustrates pressure potential. And pressure potential is also known as, well, it refers to hydrostatic pressure, which is really mechanical pressure. Okay, is pressure being placed on the membrane, on the semi-permeable membrane, pressure being exerted due to weight, let's say. In this case here, this column is increasing in height. You're going to get greater weight of water, or rather of fluid, or on this side. And eventually that's going to constitute hydrostatic pressure. We've got hydrostatic pressure being actively, or mechanical pressure being actively exerted on this side of the column by somebody pushing a piston down. And that's going to affect how water crosses the membrane from an area of high osmotic potential to an area of low osmotic potential. If we look here, we see that this area of lower osmotic potential, well, we're getting no net crossing of water across this membrane because the hydrostatic pressure, the pressure potential, in addition is counteracting the osmotic potential, right? So it's basically we're exerting force on this column, and water can't cross because it equals the osmotic potential, right? So pressure potential equals the osmotic potential. The terminology could be a little bit better worked out, but this is a constant problem in biology. We know what we're talking about, so we don't really worry too much about anybody else. But remember, pressure potential, without any reference to osmosis, is hydrostatic or mechanical pressure. It's not a matter of the concentration of the solute in a solvent.
Professor?
Yeah?
One student was just wondering what the piston in the diagrams was supposed to represent. Is that just supposed to represent pressure potential?
Yeah,
Okay.
it's mechanical pressure.
Mhm.
Okay, somebody's actually leaning on that, exerting some force to push the water back, or the fluid back down against the tendency of water to move across that semi-permeable membrane into the right-hand side of the U-bend, or the U-tube. And it's just sufficient so that there's no net movement of water. Okay, so mechanical pressure will counteract osmotic potential, or pressure potential will counteract osmotic potential. Now, water potential is the sum of osmotic potential, pressure potential, gravity, all sorts of things. Okay, so if we're going to use water potential to describe any of these scenarios, well, here we've got tension being exerted on the left-hand side. We're actually pulling water out of the area with less osmotic potential into the area with greater osmotic potential, which is counter to the way it would go if you weren't exerting this tension. So we have to add all these things up, which I'm not going to require you to do. I just want you to be aware of this, that water potential is a sum of, as far as we're concerned, summing mainly hydrostatic pressure or pressure potential and osmotic potential in determining how much water is going to, hang on,
just so a
how much
student
water is going to flow across the membrane. Sorry, take it.
It's fine. Just so a student is asking, so in that sort of way, does osmosis itself actually exert a form of physical pressure?
Oh yeah, you could consider it that. Well, the result of osmosis, like look here again, you've got a net movement of fluid into this compartment here, all right, and the column is rising. Now gravity is going to exert an effect on that. You're going to get actual mechanical pressure being exerted on the membrane here because of the weight of this extra fluid flowing in here. Okay, so that's a form of well, it's sorry, I'm putting that badly. When we talk about osmotic pressure, we talk about it in pascals, okay, or kilopascals or megapascals, the unit of pressure. So yes, it's considered to be a pressure. All right, think of it that way. And you'll get, as I said, you'll get more of this when we get into the botany section. He will talk about psi, which is a Greek letter, which is used to determine to denote osmotic potential. And of course, in plants, gravity is going to be far more important, at least in the really tall ones, in determining water potential. But yes, sorry, I got ahead of myself. That's osmotic potential should be considered a pressure. Okay, this osmosis is the tendency to move, osmotic water to move across that semi-permeable membrane. Osmotic potential is the physical effect of that. Are we okay now?
I guess some students are just a little confused about how volume can be negative.
The volume isn't negative, or you shouldn't consider it that way, but the volume can have a negative osmotic potential.
Okay.
Does that make it clear?
Yes.
All right, are we okay? I'm sorry if that slipped through. I didn't, no, volume, negative volume, boy. Yes, that would be strange, wouldn't it? Okay, so that's the basis of osmosis. Sorry, these are the basic phenomenon that we have to consider with animals: osmotic potential and pressure potential. And we can talk about water potential as well. Okay, we can sum them, but as I said, we're not going to do that in any sort of quantitative sense. I think with the plants, you will see that. But here we're talking about general phenomena. So everybody okay with this? So let's just go through it again. Okay, here's a semi-permeable membrane, and there's a volume with a higher solute concentration than this volume with the lower solute concentration, and the semi-permeable membrane separates them. The only way from here to here is through the semi-permeable membrane. All right, so what's our net water movement? Well, this has got low osmotic potential. This has got high osmotic potential. So of course, we're going to get a net movement from here to here. The arrow's bigger. There'll still be a reverse movement. Again, remember, it's easy to lose sight of this, but remember that any diffusion, whether of water or of solute, is caused by random movements of the particles involved. All right, and a random movement can be in any direction. So most of the better part of the random movements tend to be in this direction because of the lower solute content. But they can also reverse. You can also have water moving back. It's just the net movement of water is in this direction. Okay, so that's the basis of it. Now that's going to be important for understanding osmoregulation. So a little bit about osmosis in the living cell. And I know I've talked about this before, but I'm going to refine what I said. So there's, oh dear. What's the significance to animals? I'll say a little bit about plants here, but not too much. There's a cell in fresh water. It's got a high osmotic potential. The cell has a lower osmotic potential. Of course, there's all sorts of stuff dissolved in the cytoplasm. So we're going to get a net movement of water into the animal cell. And that's, let's be precise, that's an animal cell. And that's going to cause the cell to swell, which can be lethal. So we can get, if the cell is not in an isoosmotic environment, it'll shrink or swell. Right? If it's not isoosmotic with its environment, you're going to get swelling, which will be potentially fatal, or shrinkage, which also is as well, unless the cell does work. So isoosmosity with the environment is extremely important for animal cells. Plants, well, they're a little different, okay? We've got a cell wall, which, oh, there it is, okay? So in fresh water, you're going to get net movement of water into the cell until the point where the cell is exerting sufficient pressure, sufficient hydrostatic pressure against the wall. Okay, this is a matter of pressure, again, mechanical pressure being exerted against the wall, and the wall, an engineer will tell you the wall pushes back. Well, it resists the wall, it resists the pressure. Okay, and that prevents further influx of water. So we get a cell at steady state. It's turgid, it's bracing itself against this wall. And of course, that's extremely important to plants. And that turgor will prevent further influx of water. So plants are a bit better defended against a hypoosmotic or a hyperosmotic environment. Well, not so much a hypoosmotic environment. Sorry, they're a bit more well defended against a hypoosmotic environment than animals are. Not so much hyperosmotic. They'll still lose water to that. But this is important, of course, in plant structure and holding plants upright and maintaining a constant internal environment. Everybody with me? Okay, we all right with this?
Some students are kind of confused on what side of the cell the high potential is on. Is it always on the side with higher solute concentration or lower solute concentration?
High osmotic potential?
Yeah.
It'll be on the outside.
On the outside.
As a rule. Okay, now this, we're going to have to qualify this quite a bit because animals live in all sorts of different environments. Right? But generally speaking, unless the animal is dumped in really concentrated brine or something like that, in which case it won't last very long. The outside has will have a, actually, let's leave that till I get to variations in the environment, okay? For the time being, in this particular case, we're saying fresh water here has a high osmotic potential compared to the inside of the animal. Okay, so, or the inside of the animal cell. So water is going to tend to move into it. Are we all right with that?
Yeah. So just in general, water will always want to go from high potential to low potential.
High osmotic potential to low osmotic potential. Yes.
All right. That's it for questions.
And water potential is going to affect that. Okay. Hydrostatic pressure can work counter to it. So that's osmosis. Now we've looked at diffusion, we've looked at osmosis. These are both ways of getting fluids around inside the body. Okay, now we have to look at bulk flow because this is also important. And bulk flow is basically, it's the transport of fluids around the body, and generally speaking in vessels like blood vessels. And it requires the application of hydrostatic pressure. Okay, you generally have a heart, which is applying pressure to the fluid inside these tubes. And that's what we can see here. This doesn't show quite what I want, but it was in your book, so I grabbed it. But we can see that the blood flow rate is constant. But if we look down here, we can see that the blood vessels branch and branch again and branch again. Okay, now the total cross-sectional area, don't worry about remembering this, I'm just demonstrating something here. The total cross-sectional area increases quite a bit from here to here to here. All right, there's the total cross-sectional. That's in square centimeters. That means that the blood's flowing through a much greater, effectively a much greater volume here than it is here. Here it's really highly pressurized and moving fast. Here it's meandering at relatively low pressure through a whole lot of little tiny vessels. Now I'm going to talk more about this when I talk about the circulatory system. But note here, the velocity drops considerably as it goes into all these little vessels, and so does the pressure. So hydrostatic pressure is important in getting material around, getting fluids around inside the large multicellular, complex multicellular body. And that's going to affect the way that you get exchange between the fluid in the bulk transport system and the extracellular fluid. Now, this is in closed circulatory systems. That's something I haven't defined yet. A closed circulatory system means that the transport fluid is confined at all times to the vessels or to the heart, but it doesn't circulate freely through the tissues. The cells aren't bathed in, not directly. Okay, so you don't have well, you do have leakage. You don't have the blood vessels sort of ending and letting the blood flow out of them into the tissues, which you do see in open circulatory systems. Situations different there. But I want to talk about closed systems here, closed circulatory systems like we have. So bulk flow is going to be affected by things like osmosis and hydrostatic pressure. And an example from animals, actually this is human, but it'll do. I mean, the figures are human, and don't worry about them, okay? This is just to demonstrate something. If you've got a capillary bed, there's an arteriole. Now, these are blood vessels that carry blood. They're they're these guys here, all right? They give rise to capillary beds. And there's a venule. Capillaries converge to form venules. So blood comes from an arteriole, and the arteriole branches out madly into all these little tiny capillaries, which are really thin-walled. And these little tiny capillaries eventually all come back together to form another larger vessel, a venule, which carries blood away from the capillary bed. So blood's coming in at this end and going out at this end. And we note that the blood velocity has dropped a lot, and its pressure has dropped a lot too. So there's our arteriole, and there's a capillary, highly schematic, and there's a venule. So blood's going from this direction through the capillary bed into the venule. Ignore this. It's important if we're looking at human physiology, but no, we're not. Okay, so the pressure potential on the upstream side, on this side, is high. It's capillary, well, they refer to capillary hydrostatic pressure here. I'd rather you didn't worry about that too much. Just listen to what I have to say about it, and it'll make better sense. This is a little bit more complex than we need, but it was all I could find. Anyway, hydrostatic pressure here, although it's dropped a lot from the heart, it's still high. It's about 35, 40 millimeters of mercury. Anyway, that's going to exceed the osmotic potential of the extracellular fluid. Essentially, fluid and the solutes it carries are squeezed out of the capillaries into the extracellular fluid. Now, this is really important. You have a lot of turnover between your extracellular fluid and your blood. It takes place all the time. I'll say more about that when we talk about about excretion. So you've got this fluid and solutes being squeezed out, and then you get over to here. Well, you're losing solutes, you're losing water in this blood. There are big molecules in there, blood proteins of various sorts, that don't get squeezed out. They're too big to get through the capillary wall. So as the blood travels through the capillary, its volume decreases, but the concentration of these big proteins and some other molecules increases. So the blood becomes, its osmotic potential becomes less and less relative to that of the extracellular fluid. And as a result of that, we get reabsorption. Water gets drawn back into the circulatory system from the extracellular fluid because of the change in concentration of the blood. So hydrostatic pressure sends it out. Hydrostatic pressure isn't so high in the tissues, there's nothing to exert it. There's no heart working on the pressure of the fluid in the extracellular fluid, nothing comparable rather. So the concentration of solutes increased in here. As a result, it's got a lower osmotic potential than this extracellular fluid, and the water gets drawn back in, and some things that are dissolved in it as well. So that's where water potential and osmotic potential are important in bulk transport. Now, this example was for vertebrates, right? But I think you'll see something like that in any type of animal that has a closed circulatory system. As we're going to see, they don't all have that. We're going to talk next week, I hope, about closed circulatory systems versus open circulatory systems. But I wanted to bring that in to show that osmotic potential and water potential are important in transferring solute and water in the body as a whole. So you've got a long-range transport system, but it's still relying upon a long-range bulk transport system. It's still very much affected by the solute content and the hydrostatic pressure being exerted on it. So osmoregulation in animals, finally. We've dealt with ways of moving solute and moving water around the body. Only one of those was a long-range method, okay? The bulk transport system. But the others are very, very important as well. I want you to remember that. And I did talk about diffusion as being important in getting things in and out of the body at the organismal scale. We'll talk more about that when we talk about the respiratory system, certainly. Okay, so remember I said not too long ago, environments.
Actually, Professor, we have a question about the previous slide.
About the what? Oh, yes.
Why would it be exceeding the hydrostatic pressure of the extracellular fluid and not of the capillary fluid?
The volume's decreased. You've had a lot of water being squeezed out. This figure actually is relevant, 24 liters a day. Okay, 24 liters a day of blood plasma gets forced out by hydrostatic pressure, mostly hydrostatic pressure from the circulatory system, from the capillary beds into the extracellular fluid. And you reabsorb 20.4 liters per day, okay? Due to the fact the volume has dropped in here and the solute concentration has gone up, relatively speaking, because, as I said, there's large proteins in there. There's immunoglobulins and so on and so forth, all these big transport proteins, immune system proteins, all these things in there that they don't go out through the wall. So they stay in solution. And this blood becomes essentially hyperosmotic to the extracellular fluid. Does that make it clear?
I'm not seeing anything in the chat. Oh, sorry, someone's speaking. Go ahead.
Oh, yeah, just because it's probably easier if I just talk than typing it in quickly. But so is the hydrostatic pressure then during that reabsorption part or just before the reabsorption, is the hydrostatic pressure higher in the the extracellular fluid or the blood in the capillaries, I guess?
It'd be higher in the extracellular fluid, but I didn't really want you thinking about that. Okay, you've got to have some hydrostatic pressure in there just because you're cramming fluid into a finite volume. And you're increasing that amount of fluid in that finite volume. When I say a finite volume, it's the finite volume of the tissue. All right, so you are cramming more fluid in. That's going to drive the hydrostatic pressure up. Is the hydrostatic pressure lower in here? It's yeah, a little bit. If you look at the numbers here, 18 for the capillary, and 25 for the 25 for the blood, and 18 for the capillary. No, that's not relevant. Sorry. Yeah, it'll be higher. But osmosis is the main thing that's driving the movement of fluid and solutes back into the blood.
Okay, I see. Okay, thank you.
Okay. Where was I? Okay, now I want to, oh, good. Now I want to talk about different environments and how animals osmoregulate. We've seen how you move fluid around the body in various ways, short range and long range. Now, how do animals deal with the environment? Well, we're going to look at fresh water and salt water and look at how they relate to the extracellular fluid, the plasma. And that's a general term I'm going to use. Extracellular fluid, that could be hemolymph, it could be plain old interstitial fluid among the cells of the tissue, it could be blood plasma. Let's just refer to it as plasma for the time being. Now, there are two routes, generally speaking, animals can follow. There's osmoconformation, or they could be osmoconformers or osmoregulators. What's an osmoconformer? Well, there's an animal. There's the concentration of solute in the environment. This is an aquatic animal. Sorry, I should have said that right away. Terrestrial animals are a different matter. Okay, they don't live in an environment with which they can freely exchange ions or water. All right, they live in the air. So we'll leave those off the table for the time being. So there's an aquatic animal, and there's the solute concentration of its environment. It's plastic. And there's a cell in the animal, and there's the solute concentration inside the cell, and there's the solute concentration of the extracellular fluid or the plasma. So what does an osmoconformer do? Well, it allows the osmotic strength of the cells, that is the concentration of solute in the cells, to be the same as the concentration of solute in the extracellular fluid or plasma, to be the same as that in the environment. So it's like this. After all I had to say about how animals spend energy regulating their internal solute content and water volume so that the cells don't have so much work to do, I come around and say something like this, that there are animals that just don't bother. Well, in certain types of environments, they get away with it because they're living in environments that, osmotically speaking, don't change very much. Marine environments in which the solute content of the seawater stays the same all the time. And they are still not doing quite the passive thing that it looks like. Solute content, yes, is the same here and here as here, but the ions involved can be quite different. What are environments? Examples? Well, marine invertebrates, mollusks, some arthropods, jellyfish, they're hopeless, they can't regulate anything. Cnidarians in general. Those are examples, and a lot of little minor phyla that are totally that are marine. Starfish, not starfish, sorry, echinoderms, hagfish, and elasmobranchs. I'll talk more about what elasmobranchs do. But most of these organisms, like I said, aren't quite as passive as it sounds. They're changing the concentrations of particular ions inside the extracellular fluid, and by extension, the intracellular fluid. They're maintaining the total osmolarity, the same as the seawater. And then there are osmoregulators. And again, there's an aquatic animal, and look how thick its outer layer is. And what those do, oh, sorry, there's its environmental solute concentration. There's the cell, its solute concentration, the extracellular fluid or plasma, its solute concentration. Well, they adjust the osmotic strength of the extracellular fluid, the plasma, to match Y, okay, to match the osmotic strength of the osmotic potential of the cells, the intracellular environment, and they protect this whole array from the external environment. They have a thick outer coating of some sort, and prevent the free flow of water in. So Y equals X, but does not equal concentration of Z. Most freshwater invertebrates do that, things like aquatic insects. Some don't. Things like freshwater clams are pretty bad at it. And most vertebrates do this as well, with the exception of hagfish and elasmobranchs. Oh, I should have mentioned elasmobranchs. If you haven't, I think I mentioned that when I talked about the diversity of animals. But elasmobranchs are sharks, skates, rays. That's about it. Chimeras. So, and hagfish, of course, are extremely basal craniates. They don't even have vertebrae. Oh, damn this thing. Anyway, what's happening? What do things have to deal with in different sorts of environment? Now, we're not talking about osmoconformers versus osmoregulators here. We're just talking about the challenges different environments pose to any animal. We got a terrestrial animal there. There's its cell, which appeared much before the animal does. There's the animal. Okay, it's in a dry environment. Water is going to potentially leave the cell into the extracellular environment and leave the animal as a whole. Terrestrial environments, more on them later, but a much different proposition from life in water. You're not surrounded by water. You can't reliably find it in your environment, so you have to worry about drying out, basically. And this tendency of water to leave the cells into the extracellular fluid and then leave the body, or the other way around, leave the extracellular fluid, and as a result of that, water is drawn out of the cell, a constant problem. Terrestrial animals have to regulate their solute content and their water content quite precisely. They have to do a lot of this. Aquatic animals, well, in marine environments are basically hyperosmotic. Marine, again, this is not osmoconformers, but well, if you're an osmoregulator, a marine environment is hyperosmotic. Okay, it's saltier than the extracellular fluid. So water will leave the extracellular fluid for the environment, and as a result, leave cells into the extracellular fluid if you don't do something about this. Now, you could be an osmoconformer and maintain the same osmicity or the same osmotic potential as your environment does, and then maintain the same osmotic potential in the cells as in the extracellular fluid. That's one way you can be an osmoregulator, in which you have to work. But you also have to worry if you have a different osmotic potential of the extracellular fluid of the entry of salt. Remember, I'm using salt as a shorthand for ions here. Okay, now we usually think of them as sodium chloride. We usually think of ions as sodium chloride. Those are the biggies, but it's things like chloride as well, and potassium as well, and calcium, and all sorts of other ions. So you will get a net influx of ions into the extracellular fluid as well if you don't do anything about it, unless you're an osmoconformer. If you're in fresh water, well, you have to worry about being flooded. The environment is hypoosmotic to you. Hypoosmotic, sorry about that. So there's more water outside than there is inside. Concentration of solutes is higher in the extracellular fluid in the cell, so you're going to get flooding of the extracellular fluid and thus flooding of the cells without any sort of regulatory action. And you're going to lose ions to the environment as well if you're leaky, if you allow that sort of thing to happen. And marine animals have to deal with this. They are surrounded by water, and also, being gill breathers, have to move large quantities of water across the respiratory surfaces. So the potential for water gain or water loss or ion gain are really high. Something has to be done about that, unless you're an osmoconformer. So what do we see with aquatic organisms? Body fluid osmolality varies quite a bit. This is a tilde, okay? It doesn't mean negative 1,000. It's not like osmotic potential. That's about 1 osmol per kilo. The osmolality is 1 for sharks and rays and for marine invertebrates. Now, remember what I said. We'll look at sharks and rays in a bit more detail shortly. For marine invertebrates, just remember that the actual levels or the actual concentrations of particular ions might be quite dissimilar from the seawater, but you add them up and you get a similar osmolality. of the extracellular fluid. Marine
Professor?
Yeah.
Sorry, one student is just a little confused about how salt getting into the body relates with cell membrane and semi-permeability.
Hang on. There are membranes that will allow ions to pass through. I mean, they might not get through very effectively or very easily. Remember that certainly with regard to sodium and chloride, you've always got sodium and potassium, sorry, you've always got diffusion channels that are there. And although it may be difficult for a charged ion to diffuse across a cell membrane, it's not impossible. Okay, and if you're immersed in water and there's a high concentration of, let's say, sodium outside you and you want a lower concentration inside, you've still got a concentration gradient. It can still happen. And also, ions have physiological uses inside the body. So they have to be manipulated. They have to be moved. No, actually, that's irrelevant. Does that make better sense, though? I mean, you know, we're not, I've made it sound like membranes are like completely invulnerable to certain things, but that's never quite true. Things will leak through, and any leakage is going to put you, is going to change the state between the outside and the inside.
One student had a follow-up question, just kind of a quick summary.
Yep.
I think you talked about last time, but what is the difference again between osmolality and osmolarity?
Osmolality is the number in moles, okay, of a solute. So did I say osmolality? Yes. The number of solute particles, we're talking about moles, that's the usual way chemists measure it, dissolved in a kilogram of solvent, okay, a kilogram. Osmolarity is the number of solute particles, or the number of solute particles present in a liter of solvent. And the reason we talk about osmolality is because volume of a solvent can change with temperature. All right, so that's going to change the actual concentration. In other words, you're not standardizing it, whereas weight isn't. Weight stays the same no matter what the temperature is. And chemists have helped us out by making these words differ by one letter, which is something that happens in biology an awful lot. It really is irksome. Does that help?
Yes.
Good. Okay, where was I? So we got a lot of variance in osmolality among various sorts of aquatic organisms. So there's freshwater teleos, mammals, birds, and reptiles. Well, they're not aquatic, but all right. Note that it's similar to marine teleos, or well, between marine teleos and freshwater teleos. And we have freshwater invertebrates, which we're not going to talk about anymore, but they have their own tricks. What we're going to talk about mainly now is vertebrates. And we're going to talk about sharks and rays, marine teleos, and freshwater teleos. And I'll say a little bit about terrestrial organisms in general. So we do have some that are isoosmotic with seawater, and remember, it's the number of solute particles, not the identity of them. All right, the makeup of the extracellular fluid ionically might be quite different. Well, not hugely different, but different from seawater. But the number of particles is the same. And the reason they get away with this is because the environment is osmotically stable. It also limits them. All right, we have no estuarine starfish, for instance. We have no freshwater squids, nothing like that. They don't seem to be able to make the transition to freshwater or even to estuaries, where you get a lower salt content in the water because fresh water is flowing into it. So they are confined to marine environments. Or if they do enter freshwater environments or brackish environments, they're generally really heavily armored. There isn't a lot of exposed epithelium, a lot of exposed body surface across which ion exchange and water exchange can take place. So think of things like freshwater clams or crayfish. Anyway, let's look at marine bony fish first. Okay, that's marine teleos. That's these guys. Can't remember if I told you what a teleost was. Anyway, they're hypoosmotic the environment. They've got a lower number of solute particles per unit mass of extracellular fluid. So they lose water and gain ions, particularly through the gills. All right. Water's going to leak out. Ions are going to leak in through diffusion. And what they have to do is, first of all, they have to drink. They are obliged to pass a lot of salt water over those respiratory membranes. There's no choice in the matter. They've got to get oxygen. They're going to lose water that way, but they drink to offset it. So they drink seawater. So this is one case of fish drinking like fish. And they have what are called chloride cells in the gills. And these are cells that have a lot of pump proteins, the proteins that actually move ions across cell membranes. And these are pointed out, okay? So they remove sodium and potassium and chloride ions from the extracellular fluid, and they pump it out into the water. So these fish are basically breathing ions out. And they also have kidneys, and they produce a small amount of urine. They conserve as much water as they can in the kidneys, producing a small amount of concentrated urine. There's a lot of ions that are pumped into that. And also they get rid of solute in the feces. So for fish, for marine fish, it's a matter of preventing water loss and preventing ion influx by drinking a lot of water, even though it's salt water, and getting rid of those ions by means of the kidney, by means of the chloride pumps, or chloride cells rather, and in the feces. That's freshwater, marine bony fish. Now, freshwater bony fish, like this bass here, they're hyperosmotic. They're saltier than the freshwater that they live in. Of course, freshwater is going to vary. In nature, you're always going to get some things dissolved in it, but still, the fish's tissues are hyperosmotic to that water. So they're going to lose ions and gain water. They're going to have water flooding in through osmosis, and they're going to have ions leaving the gills by diffusion. Okay, because the water, there's more water outside than there is inside. There's a greater osmotic potential outside. So they don't drink. These are fish that don't drink. They produce a lot of urine. Their kidneys are working all the time to produce a lot of dilute urine, get rid of that water, but conserve ions. And they have to replace these ions by actually pumping ions in, it says salt here, but that's ions, from the water against the concentration gradient. So they've got those chloride cells, again, that work in reverse this time. They're pointing inwards, and also from their food. So they tend to eat salty things when they can get them. Chondrichthyans, sharks, rays, and skates, they're isoosmotic to seawater, but they have lower concentrations of all of these ions than seawater, and they maintain isoosmicity with the seawater by really increasing the concentration of urine or urea in the tissues. So they got a lot of urea in there. They get some water and salts from their food. They're flooded by seawater. Okay, it moves right in through the gills. They have nothing like those chloride cells, but they have a rectal gland. Sorry, I'm getting ahead of myself. They still got to deal with this inward diffusion. So they've got this rectal gland, which is a little sort of an adjunct off the intestine, quite close to the end, and it's got cells which secrete a highly concentrated salt solution. So basically, those cells are pumping ions out of the extracellular fluid into the lumen of the intestine, and it's lost in the feces. So sharks have a very different way of handling the problems of living in salt water than teleosts do, than bony fish. Why is this? I've yet to hear a good explanation.
Professor?
Yeah.
One student had a question about the two fishes in the previous slide. They would just want to confirm if those examples were supposed to be osmoconformers or osmoregulators.
Osmoregulators.
Okay.
Sorry, I should have said that. I'm not going to say much more about osmoconformers. The shark is our example of an osmoconformer, such as it is. Okay. Obviously, a very different chemical composition to the extracellular fluid in the seawater. same tonicity, same osmolality. So this is an example of a vertebrate thermal osmoconformer. I'm not going to say very much about, oh, bloody hell. It's almost time to go. I'm not going to say anything much about invertebrate osmoconformers other than I already have. So that is all, folks. I've got to run. I've got another class in a few minutes and I have to get ready for it. Do we have any other questions? Okay, let's just have a look at the next slide. Land dwellers. Okay, that's a good place to start on Friday. Okay, if that's it, I'm going to kill the meeting. Give me a minute here. So we'll stop sharing here.
Professor?
Yeah.
Could you just reconfirm what osmoconformers are? Conformers are?
They maintain the same solute concentration in the intracellular water, in the extracellular water, as the environment. So they're not. And when I say solute concentration, that does mean that the solute, the actual solutes in the intracellular water and in the extracellular fluid are not necessarily the same as in the water around them. Okay, they can be quite different. The example there was the shark. It's got a high concentration of urea. The ocean isn't full of urea. That's a side product, a breakdown product from metabolizing nitrogenous waste.
Yeah.
But the osmolality of the shark tissues are the same as the osmolality of the ocean water.
Yeah. Some students were actually asking if you could go back over the last 2 slides from your presentation.
This will have to be really, really quick, guys. I do have to go.
Yeah.
Last 2 slides.
Yeah.
This one?
Yeah, the one before this with the fish.
Just a minute.
Okay.
This?
Yeah. So just hang on to this one for a sec or two. All right. Okay, and then on to the last slide. A different student needed that one.
Be quick.
All right. 5. Mute myself.
Are we good?
Is that good? Did you get what you need? I'm going to assume so.
Yeah.
We've got time constraints.
Okay. See you a little later, guys.
See ya.
Bye.